We will begin our journey of human development at ovulation. Ovulation releases the secondary oocyte, which is picked up by the fimbriae and taken into the fallopian tube, where it waits for the sperm. Once the sperm arrive, they head towards the egg to both sides of the fallopian tube. When the sperm reaches the egg, it must penetrate several barriers in order to enter the egg. And these barriers are the corona radiata, which are a circle of cells around the secondary oocyte, protective layer, and then we have a clear layer called zona pellucida, and then we have the cell membrane. In order to penetrate these barriers, the sperm releases enzymes called acrosomal enzymes that are in the acrosome portion of the sperm. These enzymes help to digest these three barriers, the corona radiata, the zona pellucida, and the plasma membrane, in order to enter the egg. So we see just one sperm entering. There are many other sperms trying to enter the egg at the same time. How does the egg stop the other sperms from penetrating and bringing more genetic material into the egg than is needed? We only need one haploid set of chromosomes to enter the egg. The egg accomplishes this by carrying out what is called the fast sperm block and a slow sperm block. So let's see how that happens. As soon as the sperm penetrates the egg, sodium channels open and sodium rushes in, thereby depolarizing the egg cell membrane. This depolarization stops other sperm from entering. This is called the fast block, where the egg immediately blocks further entry of sperm into the egg. Next, we have the slow block. When the egg cell becomes depolarized, calcium channels open and calcium rushes into the egg. We know that calcium can cause exocytosis. The egg contains cortical granules that are arranged in a circle. When the calcium enters, these cortical granules undergo exocytosis and release a fluid that surrounds the egg cell membrane, causing a physical barrier for the other sperm. We call this physical barrier the fertilization membrane, and this is the process of the slow block against polyspermy, because we don't want more than one sperm and one set of haploid chromosomes to enter the egg.